Hope you all are doing well. Welcome to today's live session on Test and Discussion 24-25 for the subject Physiology. I am Dr. Kanimuri Sadasivam, Professor of Physiology and your mentor for Physiology here at Medico. At the end of today's live discussion, I honestly hope that you will approach all your physiology questions and questions based on physiological concepts with ease and confidence. Without wasting much time, let's proceed with the questions. So here we have our first question on the topic general physiology. We have a 50-year-old male patient who presents with symptoms of fatigue, night sweats, and an enlarged spleen. Bone marrow biopsy reveals high number of abnormal cells. The bone marrow biopsy reveals high number of abnormal cells. Further tests show that there are cells accumulating in the G2 phase of the cell cycle. Which of the following regulatory mechanism is most likely disrupted in these abnormal cells? So to answer this question, you should have a sound knowledge of the cell cycle, what are the various phases of cell cycle, and what are the regulatory mechanisms that is available, and what are all the various checkpoints where the cell cycle and what is cell cycle. So as you can see here, cell cycle is nothing but series of events So cell cycle is nothing but the series of events that cell undergoes as it grows and matures. Okay, so cell cycle is a series of events that takes place in a cell as the cell grows and matures. So what are the series of events in the cell cycle? Mainly we have two events. One is interphase, the other is mitosis. Two major events are interphase. 90% of the cell cycle events are interphase. Remaining 10% are the events that occur during the mitosis phase. Please remember, the events that happen in the cell cycle are a very tightly regulated process. Okay, and there are various checkpoints which make sure the cell cycle proceeds without any damage. Okay, so first let us see what are all the different phases in this interphase. So under interphase, the first we have is the G1 phase. So the first phase is the G1 phase, also known as the gap 1. G1 stands for gap 1. So what happens in this G1 phase? G1 phase is basically the phase of cellular growth. So the cell grows, it produces new proteins, and also the cellular organelles are formed in this phase. What are all the regulatory mechanisms, that is the regulatory proteins, the enzymes and genes that are available which checks this phase of the cell cycle? It is mainly P53 gene and RB protein. Okay, now the cell proceeds from G1 to S phase. S phase is also called as the synthesis phase. S phase is also called as the synthesis phase. What does it synthesize here? DNA replication happens during this S phase. But before the synthesis phase, as the cell proceeds from G1 to S phase, there is a checkpoint. There is a checkpoint. There is a checkpoint for the cell from G1 to S phase. What does it check? Whether the DNA is intact or not, whether the DNA is undamaged or not. What is going to do this checking? Mainly the P53, RB proteins, and also additionally the cyclin D. All this is going to check whether the cell, the DNA is undamaged or not. Okay, now it is passing through the G1 to S checkpoint and the cell enters the S phase. In the S phase, as I already told you, synthesis phase, DNA synthesis or DNA replication is happening. And what are the regulatory mechanisms that is available in the S phase? It is mainly cyclin E and cyclin dependent kinases. For S phase, it is cyclin E and cyclin dependent kinases. Next, the cell from S phase is moving to the G2 phase. G2 phase is the phase that the cell prepares itself for mitosis. G2 is the phase where the cell prepares itself for mitosis. So here what happens again, most important event happening here is again check for the DNA. In the mitosis phase, again there is a checkpoint, that is when the cell DNA mitosis is happening, when it is preparing for the mitosis, we have to make sure that the DNA is undamaged. So there is another checkpoint at this G2 to M phase. So one checkpoint was there at S1 to G1 to S phase. The another checkpoint is there at G2 to M phase. We have another checkpoint where what are all the regulatory mechanism that is available? P53, cyclin B and CDK1. If you remember our questions, this is what they have asked. cells are accumulating in the G2 phase of the cell cycle. Why the cells are accumulating in the G2 phase of the cell cycle? Because there is a damage. When we look at the clinical presentation of the patient, he has night sweats, he has an enlarged spleen, he has abnormal cells in bone marrow biopsy. All this is indicating us towards a hematological malignancy, most probably a lymphoma or a leukemia. And where is the problem? The problem actually in our given patient is the G2 to M phase. In that phase, the regulatory proteins are not functioning properly. Because of that, the abnormal cells are not getting killed and they accumulate in this G2 phase of the cell cycle. That is what they have asked, if you remember the question. Okay. Now, what are all the regulatory mechanisms that is functioning in this G2 phase? P53, cyclin B, and cyclin dependent kinase 1. Finally, from from G2 phase, if the DNA is undamaged, the cell is going to proceed to the M phase, that is the mitosis phase. Mitosis phase is further subdivided into prophase, metaphase, anaphase, telophase, and finally cytokinesis. So, this is an important topic. Just to summarize, cell cycle as a series of events that take place in the cell as the cell grows and matures. What are the series of events? What is the order? It follows. The cell is first in the G1 phase, then from G1 it proceeds to the S phase, from the S phase it proceeds to G2 phase, and from G2 phase it proceeds to the M phase or the mitotic phase. In this, 90% of the events happen during G1S to G2, which is also called as the interphase, and the remaining 10% of the events happen during the M phase or the mitotic phase. Now going back to the options, as I already discussed, now what the basic question they are asking is, cells are accumulating in the G2 phase of the cell cycle. Now, which of the regulatory mechanism is disrupted here? Is it cyclin D overproduction, or is it inhibition of cyclin D, cyclin dependent kinase 4 or 6, or is it defective P53 function, or is it increased apoptosis? From the discussion on the table which you saw just now, what is the answer? The answer is defective P53 gene function. What is P53 gene? P53 is actually a tumor suppressor. P53 is actually a tumor suppressor. Once it identifies a defective DNA, immediately it activates P21. Once it identifies a defective DNA, it will activate P21, which will stop the growth of these abnormal cells and it will not let it proceed to the M phase. In this patient, this P53 is defective. So what is happening? These abnormal cells are not getting killed, they are not getting suppressed, and they are proliferating abnormally. Okay, I hope the explanation is clear. So any question that comes from this topics, you will be able to answer. Important is, what are all the various phases of cell cycle, what is the order it follows, and what are all the regulatory mechanisms that is available each of these phases. This is what you have to concentrate with regards to cell cycle. Now let's proceed on to the next question. Okay, here, what is the question? Which of the following statements about cell organelle is or are true in the context of their role in cellular pathology? So this is a question on cellular organelles. So again, this is a very important topic, high yield topic, where you have to know what are all the cellular organelles, the function of these cellular organelles, and most importantly, what is the marker for these cellular organelles. That is the thing we are supposed to know with regards to this topic. So here let us see what is the cellular organelle they are talking about. The first option is, mitochondrial dysfunction can lead to disorders characterized by impaired ATP production and muscle weakness. Is it true? Yes, actually it is true. Let us see the next option. Lysosomal storage diseases are caused by defects in Golgi apparatus leading to improper protein processing. No, this is not true. Next option, endoplasmic reticulum stress is linked to diseases such as diabetes and neurodegenerative disorders due to accumulation of misfolded protein. I don't think this is the right answer. And also, option D, ribosomal defects primarily affects the DNA replication and repair mechanism in the cell. Okay, so they have given function for each of these organelles and only one option is correct here. So again, before answering this question, let us look at the different organelles, their function, and also what is the marker for these organelles. So first is, let us look at this nucleus. Okay, what is the major function of nucleus? Nucleus is a site of DNA synthesis and also RNA transcription, right? And what is the marker for nucleus? DNA. DNA is the marker for nucleus. Then comes mitochondria. In our option A, we had mitochondria. So we all know mitochondria is the powerhouse of the cell. So it produces ATP via oxidative phosphorylation. And any defect in ATP production is going to lead to disorders which is manifesting as muscle weakness. Okay, and then the marker for mitochondria is glutamate dehydrogenase. Very important, please remember and memorize these markers. Then comes ribosome. The ribosome, what is the function of ribosome? Ribosome is a site of protein synthesis. Ribosome is a site of protein synthesis where translation of mRNA to newly formed proteins will happen. So the marker for ribosome is the high RNA content. Then comes endoplasmic reticulum. What is the function of endoplasmic reticulum? We have two types of endoplasmic reticulum. What is it? Smooth endoplasmic reticulum and rough endoplasmic reticulum. When do we call it as smooth and when do we call it as rough endoplasmic reticulum? If ribosome is attached to it, we call it as rough endoplasmic reticulum, and if ribosome is not attached to it, we call it as smooth endoplasmic reticulum. Okay. Now, the important function of the rough endoplasmic reticulum is protein synthesis. The important function of rough endoplasmic reticulum is protein synthesis. The smooth endoplasmic reticulum, what is the function? Lipid synthesis. So lipid synthesis is the function of smooth endoplasmic reticulum. Protein synthesis is the function of rough endoplasmic reticulum. And also endoplasmic reticulum is the site of oxidation of many xenobiotics because of the presence of cytochrome P450. Okay, now let us move on to lysosome. What is lysosome? The lysosome is also called as the digestive system of the cell. Digestive system of cell. Why? Because it contains many hydrolytic enzymes that causes degradation of cellular debris, pathogens, and dead and worn out cellular organelles. And what is a marker for lysosome? It is acid phosphatase. What is a marker for endoplasmic reticulum? It is glucose 6 phosphatase. Moving on to the plasma membrane. What is the function of plasma membrane? You all know. Most important function of plasma membrane is transport of substances across the cell membrane. Not only that, it also gives the structural integrity to the cell. And also it is very, very important for intercellular adhesion and intercellular communication. And what is the marker for plasma membrane? Sodium potassium ATPase and also 5-nucleotidase. Then Golgi apparatus. What is the function of Golgi apparatus? Golgi apparatus is a region where sorting of proteins happens. Sorting and packaging of proteins happens in the Golgi apparatus. The Golgi apparatus, marker for Golgi apparatus is galactosyl transferase. Then comes peroxisomes. What are peroxisomes? These are the structures where degradation of fatty acids happen. Beta oxidation is happening, and also production and degradation of hydrogen peroxide also happens in peroxisomes. The marker for peroxisome is catalase uric acid oxidase. Cytoskeleton. For cytoskeleton, what are the different cytoskeleton? Cytoskeleton again gives the structural integrity to the cell. Mainly, it is made up of microfilaments, microtubules, and intermediate filaments. Though there are no specific markers, we still have something called as vimentin, which can be a marker for cytoskeleton. Okay, and tubulin antibodies also. Tubulin antibodies and vimentin are markers for cytoskeletal elements. And then for cytosol, what is the function of cytosol? It has enzymes for glycolysis and fatty acid synthesis. The marker for cytosol is lactate dehydrogenase. So that summarizes all the cellular organelles, their functions in brief, and also various markers. Now, after this, now I think you will be able to answer the question correctly. So, among the four options, which is the correct answer? Option A. That is, mitochondrial dysfunction leads to impaired ATP production and muscle weakness. Moving on to our next question, we have a 40-year-old woman with history of chronic liver disease. So we have a patient with chronic liver disease, presents with ascites and jaundice. As a result of liver damage, the patient is having ascites and jaundice. Laboratory test reveals elevated levels of cholesterol and phospholipids. Okay, cholesterol and phospholipids are elevated because of liver damage. Increased cholesterol, elevated levels of cholesterol and phospholipids. This alteration in the lipid profile can affect the cell membrane function. Which of the following is a likely consequences of observed lipid imbalance in the cell membrane? So what they are telling is, when the cholesterol level increases, the cell membrane function is altered. What type of alteration will happen if the cholesterol level increases in the cell membrane? So that is the question. So before looking into the options, let us first have a brief discussion about cell membrane, the various proteins that are present about cell membrane, the proteins that is present in the cell membrane and their function. So what you see here is a cell membrane. As you all know, cell membrane is a lipid bilayer. Cell membrane is a lipid bilayer, and just like any other biological tissue, the cell membrane also known as plasma membrane, also contains lipids, proteins and carbohydrates. So how much is lipids? 42% is lipids, 55% are proteins, and 3% is carbohydrates. So what are the different types of lipids that makes up the cell membrane? Phospholipids, cholesterol. Okay, mainly it is phospholipids and cholesterol. Even if you look at phospholipids, there are different types of phospholipid. Phosphatidylcholine, phosphatidylglycine, etc. For us, sphingomyelin, all these are different types of phospholipids. So basically, the cell membrane is made up of protein, fats and carbohydrates. Highest is contributed by proteins, which is 55%, followed by lipids, which is 42%. The main lipids that is present in the cell membrane that makes up the cell membrane are phospholipids and cholesterol. And then finally, carbohydrates are present in a minimal 3% is made up of carbohydrate. This is the chemical composition of our cell membrane. Now, this phospholipids forms a lipid bilayer. The phospholipid forms a lipid bilayer. As you can see, how is this lipid bilayer formed? What you see here, what I am drawing is a phospholipid molecule. What I am drawing is a phospholipid molecule. This head end, the globular head is hydrophilic. This tail is hydrophobic. Okay, so we have the phospholipids which has a globular head and a tail. The head which is present, which is facing the ECF, the head which is facing the ECF or the ICF. The globular head is either facing, globular head is always facing the fluid, either on the outside or on the inside. The attachment is so that this phospholipid bilayer, how is this phospholipid bilayer formed? The head which is hydrophilic, that is, it favors water. Okay, so those ends are either towards the extracellular fluid or towards the intracellular fluid. So you can see all these globular blue heads are facing towards the ECF or facing towards the ICF. In between you have the tails which are hydrophobic, which is sandwiched in between. It doesn't come in contact with the water. Okay. So I said the cell membrane is made up of different types of lipids. One is phospholipid, the other one is cholesterol. So where do you see this cholesterol? Interspersed in between this phospholipid bilayer. Can you see? As I am marking, interspersed in between this phospholipid bilayer. That is, between the phospholipid bilayer, you can see cholesterol. Okay, so interspace between this phospholipid bilayer, you have these cholesterol molecules. Okay, so that is with regards to the lipids that is present in the cell membrane. Okay, then coming to proteins, 55% of the cell membrane is made up of proteins. Okay, this proteins. Okay, now another word about this, the ratio of phospholipid is to cholesterol. Normally, the ratio of phospholipid to cholesterol is 1:1. The ratio of phospholipid to cholesterol is 1:1. And it is because of this presence of this cholesterol and phospholipid that the cell membrane is a fluid membrane. We also call this as fluid mosaic model. It is not rigid, it is flexible. Okay, now if the cholesterol increases, if the cholesterol level increases, as they have asked in the previous, as I asked in the question we are discussing, what will happen? The cell membrane is going to become more rigid, or it is the stability of the cell membrane increases, making it more rigid. And also, if the phospholipid content increases, the permeability of the cell membrane decreases. So you have to remember two things. When the normal ratio of cholesterol is to phospholipid is 1:1. If the cholesterol increases, where do you see the cholesterol? Interspersed in between the phospholipid. Now, if the cholesterol level increases, it stabilizes the cell membrane. At the same time, it loses its flexibility. It becomes more rigid. And if the phospholipid level increases, then what is going to happen? The permeability of the cell membrane decreases. This is a very key point you have to remember. That is with regards to discussion on the lipids that make up the cell membrane. Then let us move on to the proteins that makes up the cell membrane. Okay, the proteins that makes up the cell membrane, actually we have two types of proteins. Okay, integral proteins and membrane proteins. We have two types of proteins, integral proteins and membrane proteins. What do you mean by integral proteins? Also known as transmembrane protein. So what you see here is an integral protein or a transmembrane protein. Why do we call it as integral protein? Because it spans the entire width of the cell membrane. From the ECF to the ICF, it spans the entire width of the cell membrane. That is why we call it as transmembrane protein. Okay, what are all the functions or of this transmembrane protein or the integral protein? These integral proteins serves as channel proteins. Okay, so your example, aquaporins. Aquaporin channel is an integral protein. The sodium channel, potassium channel, calcium channel, chloride channel, different ion channels are also examples for channel protein. These transmembrane proteins or integral proteins also act as carriers. Example, your glucose transporters, which is going to carry glucose into the cell, which helps in the facilitated diffusion of glucose. Okay, and also your carriers like SGLT, sodium glucose co-transporter. So they are all examples of integral protein, and they serve as ion pumps. Example, sodium potassium ATPase pump. Okay, they also serve as receptors and enzyme proteins. When it is facing towards the ECF side, okay, when it is facing towards the ECF side, they serve as receptors. When it is facing towards the intracellular cytoplasmic side, they serve as enzymes. Okay, and also finally, these integral proteins can also act as antigenic. They have antigenic functions. For example, the RBC, the integral proteins present on the RBC cell membrane for blood group. So basically, blood group antigens are examples for again your transmembrane protein. So these are all examples for different transmembrane proteins and their functions. Okay. Then I said those are the transmembrane proteins. Then I said there are also peripheral proteins. These peripheral proteins can be present on the outer membrane, that is facing towards the extracellular fluid, or they could be present on the inner membrane. If they are present on the outer membrane, they are called as extrinsic proteins. If they are present on the inner membrane, they are called as intrinsic protein. Okay. What are the functions of these proteins? Intrinsic proteins are usually enzymes. Intrinsic proteins are most of the times enzymes, and also those proteins that anchor the cytoskeletal elements. Basically, intrinsic protein helps in maintaining the cell shape. Very, very important. Intrinsic proteins helps in maintaining the cell shape. What about extrinsic proteins? Extrinsic proteins are usually cell adhesion molecules. Okay, help in attachment of one cell to the other. Now, the ratio of protein to lipid is not same. Okay, so I told the ratio of phospholipid to cholesterol is always 1:1, normally 1:1. Like that, do we have a specific ratio of protein to cholesterol? Normally, the cell is made up of 55% of proteins and 42% of cholesterol. But this protein to lipid ratio can also be different depending on the function of that particular cell. Please remember, the protein to lipid ratio differs depending upon the function of that particular cell. For example, if you look at a Schwann cell, what is the function of this Schwann cell? Myelination. The function of Schwann cell is myelination. Okay. So for myelination, what do you basically need? You need cholesterol, you need fat. So in Schwann cell, the fat content is 75%. Fat content is 75% and protein is only 25%. So if you look at the ratio, it has the least 0.23. Whereas if you look at mitochondria, especially the inner mitochondrial membrane, mitochondria is a cell which is having high metabolic activity. In a cell like mitochondria which has high metabolic activity, the proteins are contributing 75% of the cell and lipids are contributing only 25% of the cell. So if they ask you a question on the ratio of protein is to fat, mitochondria has the highest ratio of 3.2 and Schwann cells has the lowest ratio of 0.23. Sarcoplasmic reticulum, the ratio is around 2 and outer mitochondrial membrane it is 1.1. Okay, so this is also a very important topic where many questions have been asked in this. Now let us go back to our, finally let us complete our discussion by looking at carbohydrates. We saw about the lipids in the cell membrane. We saw about the proteins in the cell membrane. Now let us look at these lipids which, the carbohydrates which make up the 3% of the carbohydrates that makes up the 3% of the cell membrane. Where do you first find these carbohydrates? These carbohydrates are exclusively present on the outer surface of the cell membrane. First, very important is these carbohydrates are loosely attached to the outer surface of the cell membrane, that is towards the ECF side. Okay, and it is called as glycocalyx. What do we call these carbohydrates? We call them as glycocalyx. If the carbohydrate is attached to a protein, as you can see here, the carbohydrate molecule is attached to a protein. So we are calling it glycoprotein. Here you can see the same carbohydrate molecule is attached to the lipid. So what do we call it as? Glycolipid. Okay, the general term given to us is glycocalyx. Specifically, if it is attached to the protein, then we call it as glycoprotein. If it is attached to the lipid, we call it as glycolipid. What are the functions of these glycocalyx? This is a negatively charged molecule. The carbohydrates are negatively charged molecules. So they have negative charge, though they are going to repel other proteins which are negatively charged from attaching to the cell membrane. Then it is very, very important for selectins. Selectins are again example for glycocalyx which helps in cell to cell adhesion. Selectin is an example of cell-to-cell adhesion. Selectin is an example of glycocalyx which helps in cell-to-cell adhesion. Then they serve as receptors and they are very, very important for immune reactions as well. So now that completes our discussion on the cell membrane, the composition of cell membrane and functions of each of these components. Now let us look at this question. So what was the question? In our patient, what was the scenario? This patient has liver dysfunction. Because of that, the cholesterol and phospholipids levels have increased. If the cholesterol level is increasing, what did I say? Where will the cholesterol increase? Actually, as you can see in this picture, the cholesterol is going in interspersed. Cholesterol is getting inserted in between this phospholipid layer. So basically, if the cholesterol level increases, what is it going to do? It is going to make the cell membrane more rigid. It is stabilizing the cell membrane. Also, phospholipid is also increasing. When the phospholipid increases, what did I say? The permeability of the cell membrane decreases. Please remember these two key points. With that in mind, look at the option. Which of the following is a likely consequence or observed lipid imbalance in the cell membrane? Option A, enhanced membrane fluidity and permeability. Definitely no. Option B, decreased receptor mediated endocytosis. No. Option C, increased membrane stability and reduced fluidity. Yes. And also let us look at the next option before confirming our answer. Impaired ion channel function. Among all these things, which is the most apt answer? It is the increased membrane stability and reduced fluidity. Moving on to the next question. Which of the following statements about the cell membrane is true? Another statement on cell membrane. The cell membrane is primarily composed of a single layer of phospholipids with embedded protein. Is it a single layer or bilayer? Always remember it is a bilayer. It is a lipid bilayer. Never forget this line. Cell membrane is a lipid bilayer. So this is not the correct option. Then the fluid mosaic model describes the cell membrane as a rigid structure with fixed proteins and phospholipids. No, actually these proteins and phospholipids can travel horizontally. They cannot move from inside to outside, but they can travel horizontally within the cell membrane. Okay, so this is also not true because of the fluid mosaic model actually describes it as a flexible structure rather than a rigid structure. So this is also not the correct answer. Next, the cell membrane has a selective permeability nature, allowing only non-polar molecules to pass through it freely. No, it is allowing even small polar molecules to pass through it. Final option. So now we are left with only one option. The cell membrane fluidity is mainly due to presence of cholesterol molecules that stabilize the phospholipid bilayer. Yes, very true. The cholesterol which gets interspersed between these phospholipid molecule only helps in stabilizing the cell membrane. So the correct answer is option D. Okay. After each question, I have given the right answer and also a discussion on why this answer is right. So when you have time, you can download this and also go through these explanations so that it will help you remember and understand the concepts better. Next question. Which of the following components of the extracellular matrix is primarily responsible for providing structural support and tensile strength to the tissues? What are all the options? Glycosaminoglycans, collagen, fibronectin and elastin. All these are components of extracellular matrix. All the four options that is given here are components of extracellular matrix, but among this, the one that provides structural support and tensile strength to the tissue is collagen. Okay. Then what about the others? Glycosaminoglycans, mainly what is its most important function? It provides hydration and cushioning effect. It provides hydration and cushioning effect. One example of glycosaminoglycans, if you can recall, the very famous hyaluronic acid. So hyaluronic acid is an example of glycosaminoglycans. They provide hydration and cushioning effect. Okay, then comes elastin. What is the function of elastin? Elastin allows the tissue to stretch and recoil. Elastin allows the tissue to stretch and recoil. So where do you think we will find abundance of elastin? Aorta. Aorta is an elastic vessel, Wind-Kessel vessel, right? Why we call it as Wind-Kessel vessel? Because it is rich in, it has an elastic recoiling property. Because of the presence of which of the extracellular matrix substances? Mainly it is made up of elastin, which allows the tissue to stretch and recoil. What about fibronectin? Fibronectin facilitates cell adhesion and cell migration. Okay, so that's the discussion on different kinds of extracellular matrix substances. So glycosaminoglycans provide hydration and cushioning effect. Mainly hydration and cushioning effect. Collagen is giving you the structural support and tensile strength. Fibronectin is important for cell migration and cell adhesion. Elastin is important for stretching and elastic recoiling of the tissues. Okay. Now we have a 35-year-old patient is diagnosed with a genetic disorder that affects the function of sodium potassium ATPase pump. Because of this, he has an inability to maintain normal electrochemical gradient across the cell. Which of the following is likely a direct consequence of the defect, of this defect? So to answer this question, you should know in detail about sodium potassium ATPase pump. Okay. So first let us look at the function of sodium potassium ATPase pump. Okay. So what did the sodium potassium ATPase pump actually do? So this sodium potassium, very important is sodium potassium ATPase pump is important for maintaining resting membrane potential. Very important is sodium potassium ATPase pump is important for maintaining resting membrane potential. It is very important for maintaining the cell shape and cell volume. Okay, these two key points you have to remember. The sodium potassium ATPase pump is important to maintain the resting membrane potential, maintain the cell shape and cell volume. If this pump is not pumping properly, as it is defective in this patient, what will happen? The cell will take up water, it will swell, it will burst and the cell will die. Okay, so basic function of the sodium potassium ATPase pump, what it does, it pumps 3 sodium, 3 it pumps 3 sodium ions outside, okay, and 2 potassium ions inside the cell. Okay, so the sodium potassium ATPase pumps 3 sodium ions outside and 2 potassium ions inside. So what is it basically doing? 3 positive ions is going outside. In response to that, only 2 positive ions are coming inside. So the inside of the cell is deficient of 1 positive charge. That is why the inside of the cell membrane is more negative compared to the outside. That is how it helps in maintaining the resting membrane potential. Okay, how does it hold the cell shape and cell volume? Basically what it is doing, it is pumping sodium outside. When it pumps the sodium outside the cell, this is the cell, sodium is less inside, sodium is more on the outside. Sodium is more on the outside and sodium is less on the inside because of the sodium potassium ATPase pump. Okay. As a result of that, what happens? It establishes a concentration gradient for sodium. So wherever you see this word, it is a very important concept. When you say concentration gradient for sodium, that means the sodium potassium ATPase pump, by pumping 3 sodium ions outside, establishes a concentration gradient for sodium. That is, outside has more sodium, inside has less sodium. So what is going to happen? Automatically sodium will try to come from outside to inside. This is called as establishing concentration gradient for sodium. Okay. Now, if more sodium was present inside the cell, suppose if more sodium was present inside the cell, what will happen? Sodium is an again osmotically active substance. When sodium comes inside the cell, along with it, it will also carry water. It will drag water into the cell. What will happen? The cell will swell, it will burst. So that is why this sodium potassium ATPase pump is very, very important for maintaining the cell shape and volume. Okay, so these are two very important functions of your sodium potassium ATPase pump. Also, it is important for protein synthesis. This sodium potassium ATPase pump is very, very important for protein synthesis also. Why? Because the intracellular potassium is high. The intracellular potassium is high and this is needed for protein synthesis. So these are briefly the very, very important functions of the sodium potassium ATPase pump. Now, let us also look at the structure of the sodium potassium ATPase pump. Many questions have been asked based on the structure. So what type of pump? The sodium potassium ATPase pump is a integral protein, right? It spans the cell membrane around 10 times. It spans the cell membrane 10 times, very important. It spans the cell membrane 10 times and it has two subunits, alpha subunit and beta subunit. This alpha subunit is a larger subunit and it is the alpha subunit which has been extensively studied. Beta subunit is a smaller subunit and its physiological function has not been established. So much has not been studied about the beta subunit. All we know so far is the beta subunit on the extracellular domain has 3 glycosylation sites. Okay, so the beta subunit on the extracellular site has 3 glycosylation sites. That much we know about the beta subunit. Now coming to our alpha subunit, which is the most important one. Alpha subunit also has an extracellular domain as well as an intracellular domain. First, let us look at the intracellular domain of the alpha subunit. On the intracellular domain of this alpha subunit, it has actually 3 binding sites. One is for binding of sodium. So 3 sodium ions will bind at the sodium binding site. Another site is the phosphorylation site. What is this site? P stands for phosphorylation. Okay, phosphorylation site. And third site is the site for binding of ATP. And also, please remember what is the name of this sodium potassium ATPase pump. ATPase pump means it has inherent ATPase activity. So it also has AT, it has ATPase activity. So what will happen? The moment sodium ion comes and binds, the sodium ion and ATP binds to their respective sites. Immediately what will happen? This ATPase is going to split the ATP, leading to the formation of ADP and high energy phosphate. This is called as, this process is called as phosphorylation. Okay, as a result of phosphorylation, what will happen? This alpha subunit undergoes conformational change. It undergoes conformational change. Okay, before that, let us look at the extracellular domain. I hope the intracellular domain is clear. Intracellular domain actually contains three important sites. One is the site for binding of sodium. The other is site for binding of ATP. Third is the phosphorylation site, and also we have, since this is an ATPase, it has inherent ATPase activity which is present on the intracellular domain. On the extracellular side, it has two sites on the extracellular domain. One is for binding site for two potassium ions. The other is binding site for cardiac glycosylates like obamin, okay, digitalis, all of them. So all these cardiac glycosylates, these drugs, where do they bind? If they ask you, they are binding to the extracellular domain, not to the intracellular domain. Okay. Okay. Now, how is it? That is about the structure of this sodium potassium ATPase pump. Now, how do they function? As I said, it basically functions by phosphorylation and dephosphorylation. What is the most important function? Pump 3 sodium outside and bring 2 potassium inside. So once the ATP is bound to the ATP binding site on the intracellular domain, because of its intrinsic ATPase activity, what is it going to do? It causes phosphorylation. Because of phosphorylation, what happens? It undergoes conformational change. It undergoes a conformational change, so that now the entire intracellular domain is going to face outside. All this is going to face outside. So it is throwing the sodium ion outside. Simultaneously, what is happening? Now the potassium ion is coming inside. Okay. Then this site undergoes dephosphorylation. Okay. So once it undergoes dephosphorylation, what will happen? Reconformational change will happen. Because of this reconformational change, what will happen? The extracellular domain carrying the potassium ion which came inside will again revert back to its original position. Okay. So this is how the sodium potassium ATPase pump is functioning. Now let us look at this options what they have given. So here in our question, in this patient, the sodium potassium ATPase pump is not functioning properly. Okay, so this leads to inability to maintain the normal electrochemical gradient across the cell membrane. Which of the following is a likely direct consequence to because of this defect? Option A, decreased intracellular calcium concentration. Option B, increased intracellular sodium concentration. Option C, increased extracellular potassium concentration. Option C, decreased extracellular sodium concentration. So here, what is very, very important? Forget about the calcium. So this calcium is out of your equation. Now, even among these three, what is the function? It pumps 3 sodium ions outside. Okay. Now, if it is not functioning properly, what will happen? The sodium ion will accumulate inside the cell. So, the correct answer is option B, increased intracellular sodium concentration. So, the right answer is increased intracellular sodium concentration. When you get these questions, they might look little confusing. All you need to know is just try to recall your core concept. Once you know that core concept, apply that concept here. Approaching such questions becomes very, very easier and you will never make a mistake. Okay. That was with sodium potassium ATPase pump. We also have other ATPase pump or active transport pumps. One is the calcium ATPase pump. What do calcium ATPase pumps do? It is this calcium ATPase pump which regulates the intracellular calcium level. Okay. If this pump is not working properly, then what will happen? The calcium will pile up inside the cell. It increases the calcium concentration inside the cell. We also have the chloride bicarbonate exchanger. We have the chloride bicarbonate exchanger, which is very important for maintaining the ion balance and pH inside the RBC. Okay, then we also have glucose transporters, basically GLUT, which helps in facilitated diffusion of glucose into the cells. Okay, now let us move on to the next question. Which of the following mechanism of transport across the cell membrane does not require the input of cellular energy? Which of these mechanisms do not require ATP? Is it active transport? Is it facilitated diffusion? Is it endocytosis, or is it primary active transport? So this is a very easy topic. By now I am very sure all of you know this very well, and this is a very direct question. These are such questions where you cannot go wrong. Okay, so among these given options, which is a passive mechanism? That is what they are asking. The moment you see the word diffusion, it means it is a passive mechanism. So the answer here is facilitated diffusion. The other options, as you can see, active transport, endocytosis, and primary active transport, these are all examples of active mechanism. I want to add a word here about endocytosis. Endocytosis not dependent on concentration gradient. Okay, but needs energy. Endocytosis is a type of transport where energy is required but concentration gradient is not required. Okay, now let us briefly discuss the different types of transport mechanisms. Okay, I would strongly suggest you to go back to the app and look at the discussion of transport across cell membrane in detail, where we have dealt a lot of other topics, including what is saturation kinetics, what is competitive inhibition, what are all the factors that affects diffusion, and also the graph, concentration of the substance and diffusion graph, which diffusion happens better, faster rate of diffusion is faster in facilitated diffusion or simple diffusion. Lot of questions come from these topics. It is not possible to discuss everything here for the single question. I will briefly give an outline. The rest of the things, please go back and look into the app. This is again a very high yield topic where you cannot go wrong. Once the concept is clear, there will not be any confusion. So briefly, what is the use of transport? Transportation is basically to transport substances into and out of the cell. So these transport processes can be briefly an active mechanism or a passive mechanism. Okay, so transport could be active transport, it could be passive transport, and also it could be vesicular transport, which is actually a type of active transport. Vesicular transport is also a type of active transport. And also we have transport across epithelia. Transport across epithelium. Okay, so transport mechanisms are broadly divided into 4 different types of transport mechanism. The most important broad ones we will discuss now is the active mechanism and the passive mechanism. Whenever you see the word active mechanism, means what you have to remember, it is uphill transport. Two key things you have to remember when it comes to our transport. Active transport means it is an uphill transport. Active transport means uphill transport. That means against the concentration gradient. Just now I told you how concentration gradient for sodium ion is established by sodium potassium ATPase pump. Like that we have concentration gradient for different ions. Based on this concentration gradient only, we will the cell will decide whether it is going to move across the cell via active transport or via a passive transport. If it is along the concentration gradient, when I say along the concentration gradient, it means from a region of higher concentration to a region of lower concentration. If the transport happens from a region of higher concentration to a region of lower concentration, it is a downhill transport. You are coming down. It is a downhill transport mechanism. So when it comes to a downhill transport mechanism, energy is not required. No ATP, energy is not required. Active transport is just the opposite of passive transport. In active transport, it is an uphill transport. Uphill transport means against the concentration gradient, against the concentration gradient. When I say against the concentration gradient, means from a region of lower concentration, the substance is actively transported to a region of higher concentration. This is called as against the concentration gradient. So it is basically uphill movement. The moment when it is uphill, obviously ATP is used. Okay, these are the key points when it comes to differentiating between active transport and passive transport. Okay, as I told you, vesicular transport is a type of active transport where concentration gradient is not required. Okay, this is again an important MCQ. What type of transport is vesicular transport? It is a type of active transport, but it doesn't need concentration gradient. Okay, now what are all the, first let us see what are all the different passive transport mechanism that is available. So one is simple diffusion, second is facilitated diffusion, third is osmosis, fourth is, what is not here, is filtration. Okay, fifth is bulk flow, and sixth is, you tell me, solvent drag. Okay, so the cell membrane has 6 different passive transport mechanisms. So when you see any of this 6, simple diffusion, facilitated diffusion, osmosis, solvent drag, bulk flow or filtration, none of them require energy. Okay, let us look at each of them in brief. Simple diffusion means what? It is simply going to diffuse across the lipid bilayer of the cell membrane. Okay, here what you see is a simple diffusion. Okay, so in simple diffusion, what we are seeing here is the substance that needs to be transported. Okay, so it is there in higher concentration on the extracellular fluid side. On the ECF side, this particular substance that needs to be transported is in high concentration. Now, it is going to simply diffuse through the lipid bilayer and enter into the cell. It is simply diffusing through the lipid bilayer and entering into the cell. This type of transport is called as simple diffusion. No energy is required, downhill transport. It simply dissolves into the lipid bilayer and comes into the cell. When will this transport mechanism stop? It will stop when it equalizes, when the concentration of the substance equalizes on either side of the cell membrane. When the concentration of this particular substance, as you can see in the step 1, that particular substance that needs to be transported is in high concentration in the ECF. Then in step 2, what is happening? It is slowly diffusing and entering into the intracellular space. In step 3, it stops. Why it stops in step 3? Because the concentration of the substance on either side of the cell membrane is equal. Such type of transport is called as simple diffusion. Example for simple diffusion that occurs through the lipid bilayer, usually alcohol and gases diffused by simple diffusion. Okay. Another type of simple diffusion is also there. Here, this substance is not lipid soluble. This substance is not lipid soluble. So, what is going to happen? This substance has a, if you remember the structure of cell membrane where we saw different channel proteins, it could be for water, or those channel proteins could be for various ions, right? So, since these substances are water soluble, they are not lipid soluble, these substances cannot simply diffuse through the cell membrane. They don't dissolve in the lipid bilayer. So, they are going to enter the cell through certain channels. This is also an example of simple diffusion. Okay, I don't want you to confuse the simple diffusion with facilitated diffusion. Simple diffusion can occur via the lipid bilayer or through protein channels. So, example for substances which comes through this way, usually what are those? Yes, glucose, ions, mainly ions, amino acids, etc. can enter this the intracellular space via these channels, sodium ion channels, potassium ion channels. Okay, fine. Then we are moving on to the next type of passive transport, that is facilitated diffusion. We are moving on to facilitated diffusion. So what do you mean by facilitated diffusion? As the name suggests, something is facilitating this diffusion. Facilitating means something is helping this type of transport. It is exactly similar to simple diffusion. Only difference is here you have a carrier protein. It is not a channel. Please don't confuse. It is not a channel. It is a carrier protein. What is this carrier protein going to do? It is going to carry this particular substance that needs to be transported. Example for carrier proteins are glucose transporters. Okay, that is GLUT. Another example for these type of carrier proteins are SGLT, sodium glucose co-transporter. Sorry, only thing I don't want you to confuse here. Sodium glucose co-transporter is a type of carrier protein. Sodium glucose co-transporter, what I wrote, is a type of carrier protein, but it doesn't help in facilitated diffusion. That is why I removed it here. Glucose transporter is a type of carrier protein which helps in facilitated diffusion of glucose.